There is an increasing concern towards the discharge of synthetic colouring agents from chemical industries due to its impact on the environment. Many of these agents are immiscible in water and cause major problems when they are discharged. Analytical methods have been proposed for the detection of these agents, but no concrete procedures have been established or cited in literature.
The major purpose of this study was to design and optimize an accurate, sensitive, analytical method for the quantification of mutagenic dispersed dyes produced in textile and dye-making industrial processes. The major analytical processes used included reverse phase high-performance liquid chromatography coupled with a diode array detector. These samples were analysed against industry standards and via standard addition to samples.
Determination of these dyes was conducted on the wastewater effluent from dye-making manufacturing facility along the Cristas River in Sao Paulo, Brazil. 9 samples were collected from various locations along the river in order to identify the level and extent of contamination within the designated region. The analytes that were tested were dispersed blue 373, dispersed orange 37, and dispersed violet 93.
These are components of a black dye used in the dyeing of polyester. The non-adsorbed dyes end up in wastewater, some bioaccumulates, and thus is believed to contribute to the mutagenicity detected in the river that was analysed. 6 kilometres downstream of the textile's industrial site is where a drinking water plant is located, and thus the dye is believed to be contaminating the drinking water due to lack of treatment for non-biological material.
Other analysis methods used apart from HPLC and DAD include mass spectrometry as well as carbon and hydrogen NMR spectroscopy to determine their purity and confirm their structures.
After collection, the liquid samples are run through liquid-to-liquid extraction. The residual of this is then rotary evaporated, then dried with a flow of nitrogen gas. The resulting powders were then added to an aliquot of acetonitrile.
The stationary phase used for the separation of the analytes in the sample was a C18 reverse high-performance liquid chromatography column. The mobile phase consisted of a mixture of acetonitrile and water. The acetonitrile acted as a solvent for the organic compounds, whereas the water acted as a polar elegant.
The internal structure of the column acts as the stationary phase, consisting of a tightly packed lattice of micron-sized porous silica particles. Each of the particles has a coated surface of saturated alkyl carbon-18 chains, which exhibit nonpolar hydrophobic behavior. These properties instigate interactions with the sample, resulting in the partitioning of the analytes from the mobile phase as the mixture travels through the column.
Each of the dyes in the sample interacted with the stationary phase at varying strengths based on their structure, as some of the dyes are more nonpolar than others. These nonpolar sites on each molecule resulted in the adsorption to the stationary phase, forming partial bonds to slow elution time and retaining each analyte respectively. The order of elution of the analytes was orange dye, violet, and blue dye.
The carrier solution used was 85 acetonitrile to 15 water. In order to avoid problems with detection such as overlapped peaks, a mixture of standards of the commercial dyes was run several times with differing ratios of acetonitrile to water. By experimentally seeing which mixture gave the best separation of the dyes in the chromatograph, the ratio of 85 to 15 was chosen as the ratio which gave the best elution.
These conditions were experimentally optimized by making repeat measurements with differing running conditions and evaluating which conditions gave the best symmetry and resolution for the chromatograph, while keeping a short analyzing time.
For quality control, the collected samples were tested using standard addition with each isolated dye. This was used to eliminate matrix interferences, which is a common issue within the analysis of samples. Overall, it was found there was good repeatability and accuracy of results, as all chromatographic procedures were performed in triplicate.
The method was tested by determining the detection limit. This was achieved through spiking the water samples collected upstream from the textile runoff. This was to simulate low dye concentration levels. Good recoveries of the dyes were obtained, confirming the accuracy of the procedure.
The analytes were detected using a UV-Vis diode array detector, coupled with the HPLC. The combination of tungsten and deuterium lamps gives off a broad emission spectrum of wavelengths between 200 and 800 nanometers. Thus wider spectrum of analysis.
As the solute is eluted, The light through the flow cell is dispersed by diffraction grating into the diode array. The use of multi-wavelength detector is advantageous, as the analyte is conducted at multiple wavelengths simultaneously. Therefore, the absorption of the analytes is easily obtained.
With the analytical method described, it was determined that the water cleaning process was not efficient at removing the mutagenic dyes from the river system. However, this new method of using HPLC coupled with a diode array detector was deemed to be efficient for the monitoring of the 3 dispersed mutagenic dyes within the tested area, resulting from the textiles manufacturing effluent.
Let's go again.
Sorry, can you start again?
Number 5.
Let's go again.
Number 5.